Welcome, everyone. My name is Tim Camp. I'm director of the Stem Cell and Regenerative Medicine Center here at the UW. And on behalf of the University of Wisconsin, the Morgridge Institute for Research, with the Wisconsin Institute for Discovery and WARF, I'd like to welcome you here to the Crossroads of Ideas. And this particular event is really to help celebrate a tremendous discovery that was made here at UW that I think all of us at the university and in Wisconsin are excited about. That was the discovery of human embryonic stem cells by Jamie Thompson 20 years ago that really opened up a whole new field of research and medicine, ways to study the human body that weren't possible before, and also what we're seeing now is new avenues for therapy. So it's really an important event for us here at Wisconsin. And so I'm as interested as you are in finding out what the next 20 years are going to bring. And I think we'll have a good chance at guessing and looking forward with our panelists. But of course, there's a lot of excitement to come. So I just want to tell you about a few other things. And after this discussion, as the doors go up, there'll be stations outside that you can visit with some of our students that will show you a variety of things about stem cells, some contracting heart cells, and a few other things. So if you'd like to have some interaction with students and see some of the cells in action, that'd be a good opportunity. Also, great opportunity here. You get a bonus take-home. You get to beat the Sunday paper because this Sunday there will be an insert about 20 years of stem cell science at Wisconsin. It was produced by the Wisconsin State Journal and the Morgridge Institute for Research, and you can pick that up on your way out if you'd like to. Then I want to remind you that the next Crossroads will be on December 4th, and the topic for that will be how not to think about artificial intelligence. So with that, I'd like to introduce today's moderator. Penny Patterson is Vice President for Communications at Promega, and you'll see a gifted moderator, and she'll keep us on task and on time. So Penny, please.
Expectation there. So exciting to be a part of this event tonight. And I'll confess up front, how's the volume? Is that working? That doesn't sound, yeah, sort of sound like a little munchkin right now, don't I? I just keep talking and I'll trust that the audio guy is, oh, it's adjusting now. Yeah, or I could talk lower. Would that help? At any rate, I have to confess that I am a non-scientist from Promega. How many of you are actually scientists who are in the room? Okay, you scare me. There's too many of you. But it's a thrill to have both the scientist and the non-scientist here today. So I leaned on some of my colleagues in preparation for this evening to appreciate the story of stem cells and appreciate not only what we're going to hear about in terms of how they can really better our lives, but over the last 20 years, there's been a tremendous ripple effect from these discoveries. And I appreciated from my colleagues from BTC, some of who are here today, the BTC Institute, that Madison College has a whole stem cell technician program thanks to this discovery. From the BTC Institute, there are all sorts of classes and curriculum around cells. Oh, sounds like the audio got worse, didn't it? How's it doing? Better? Maybe I'll just talk louder. Would that help? But at any rate, a new mic would be good, right? Closer? I'm eating it right now. What do you mean? So I did want to mention that the Institute also helps with the Stem Cell Symposium in concert with the UW every year, which is pretty exciting. And also some of the ripple effects. Should I try that other mic? It's driving you crazy, isn't it? Yeah, let's try that one. Try not to get feedback. How's this? Toward the end. Okay, then we'll figure this out later. We'll have to share a lot of mics. But also at Promega, the fact that our tools are used in stem cells, and that's very exciting, and our VP of Research was sharing that story and appreciating, most importantly, how stem cell discovery has really changed and created a new paradigm in science. And of course, that's why all of you are here today, right? To hear a little bit more about what happens and what might happen in the next 20 years. So, with that, enough about Penny and Promega and much more about our scientists. We're going to ask each of them to introduce themselves and answer three primary questions. Are you ready, friends? You need to address the question that you're working on right now, the question you're trying to answer, and the goals. That moment that you knew this was your life work. Remember that spot. And lastly, personally, what drives you to keep doing work? So with that, Lynn Allen Hoffman, we'll start with you.
Thanks a lot, Penny. And I'd also like to tell all of you in the audience what an honor it is to be able to talk to you tonight about the work that not only that I've been doing, but a large group of young people started to help me do here at the University of Wisconsin. And I'd also like to acknowledge a much larger and broader group of individuals that are helping us complete the work that was started here. And this is a group in New Jersey, and it's a specialty pharmaceutical company called Mallinckrodt. And I'm just going to mention that because they're essential for us getting this technology to patients. And that's going to be what I finish with at the end, because that's what drives me. But with regard to telling you a little bit about what scientific question that I'm interested in, I have had a specific curiosity about human skin. And I started that curiosity really when I transitioned from graduate school to, after getting my PhD, and I got that in Boston, and I postdoced. That's additional training. I see a bunch of young people here, which is fabulous, I have to tell you. So that's additional training you get after graduate school. And I specifically did not want to work on animals. I wanted to work on human cells because that was most relevant. So a theme is the relevance of how I apply a curiosity I have, I guess, in the broader context. And a model system at the time that was new was how you grow skin cells. Now, I'm up here talking to you today as part of a human stem cell celebration and the celebration of how to grow human embryonic stem cells. I have worked with human embryonic stem cells in my lab at the University of Wisconsin, but what I've primarily dedicated my career to and most of my thinking to is the type of stem cells that are in all of our bodies that enable our tissues to regenerate. And one of our most regenerative tissues is our skin. And it's a simple organ. It's made up of two layers. It's made up of a dermis and an epidermis. And that epidermis is really unique because the cells, what's called tissue-specific progenitor cells, and this is a type of skin cell, reside right in that very first layer. And it produces multiple layers or different strata. And it's important to us because it constantly regenerates throughout our entire lives. All of you are going to produce and shed about 40 pounds of human skin cells. That is the dust in your house. and your pets, etc. And embedded in those, in your skin are also appendages. Your hair follicles, your sebaceous glands, even your fingernails are appendages of your skin. And those are special areas, we'll call them niche areas. In the scientific terminology, they're called niche areas, where other special stem cells reside. So I don't work on human embryonic stem cells. I've been interested in how our skin renews itself. So the question I asked was, what controls that? When it's uncontrolled, you have skin disease. And I think I was interested in it because it was very visual. you could see it. I mean, we all know quite a bit about our skin. We medicate it our whole lives. We take care of it. We see when it's wounded. We cosmetize it. We know if we've got a problem with it. And so it's just a very accessible tissue. So I've always had a curiosity about it. So how did I get to where I am? I've put a lot of human tissue samples into what's called tissue culture, where you can grow the individual cells, you disaggregate them, and then put them into a tissue culture dish and grow them. And when we do that, you can, within that population are the cells that can reproduce the tissue. They're, again, called these progenitor stem cells. And in doing that, I had a serendipitous discovery happen in my lab. I might really want to emphasize to this audience that my contribution was unbelievably humble. I did nothing, absolutely nothing. I had no hypothesis. I didn't treat the cells with anything, but I was doing an experiment. We had a lot of dishes, and in the control dish, which had nothing put into it, and controls are always important in doing any experiment. I'm looking at this group here. There was a single colony. And in that dish, my very competent technician, and she and I have worked together, I might add, 30 years. So I hired her right out of undergraduate school, and she and I still work together. And she asked me to look at this under the microscope. And again, this is a very social type of profession, I believe, actually doing stem cell work. You need to explain your observations and get feedback about what you're seeing. And I told her, well, Sandy, don't fix and stain that dish, because that colony there looks like what's called a basal cell, which is a stem cell in human skin, or that's where the stem cells reside, in that first layer. And so she put it back in the incubator, and she very fastidiously cultured these cells for a year at a very specific periodicity. And we came to appreciate that they did not do something called senesce. That is, we all have a natural number of cell replications that all our cells are going to go through before they can no longer replicate. We're mortal individuals, and this is can be demonstrated in tissue culture. And what we found in Sandy culturing these cells with a very regular periodicity was that they had acquired the characteristic of being able to be long-lived. And with that, I was convinced that they, and I'll wrap this up maybe, would be aberrant in some way. And I worked for many years, well, 3 years specifically, thinking about this the wrong way. And because I had the original cells from this particular infant. And it wasn't until I met the director of the burn unit, and this was, and I want to give credit to the burn nurse at the University of Wisconsin Burn Center, when we went to get some bandages for some experiments we were doing, actually on animals. But that she challenged me, and she said, would you talk to the director of the burn unit? And I said, yes, I'd be happy to talk to the director of the burn unit. She asked me what we were doing, and I was just wanting some bandages. And so in that meeting, I told him a lot of detail. He was not interested in the detail I was telling him because I was telling him a lot about the characterization we'd done, some molecular biology, some protein work, and he told me that I didn't know what he needed. And this is an important part of what drives me. And I recognized when he told me that he was right. I didn't know what he needed. But he challenged me in a way, and he said, would you watch me do a burn surgery? And I said, yeah. I grew up on a farm. I'd give me a call. I'd be happy to come. And about 9 months later, he did. And he gave me a call, and he said he had a 95% total body surface area burn. That was a 3rd-degree burn of a young farmer from southern Wisconsin. And I went into this surgery. I'm a PhD, I'm not an MD. I went in at 6 o'clock in the morning. I came out at 2 o'clock in the afternoon. I went down campus to a seminar. The that I attended on a weekly basis, and I was sitting there listening to the speaker speak, and I thought, what am I doing? I was a tenured full professor. I had something in my lab that might help someone, and that was my eureka moment. That's when I knew what I should be doing. And I do want to clearly express what a unique place the University of Wisconsin is because they let me do it. I was running a center at the time, a multi-college center. I told my chair that I felt that I really needed to focus on developing this in some way to see if it could help someone, this technology in some way. And I said, this is just very clear to me. I don't know why it was so clear to me, but that was indeed my eureka moment, and I'll pass this on to my other colleagues.
Right, I'm David Gam, and I'm an ophthalmologist, so I like eyes. And then my PhD was in neuroscience, and so I like the brain. And so within the eye, there is one very distinctly brain-like, or very much is the brain, tissue, and that's the retina. And that lines the back of your eye. It's like film in a camera, and it's actually a series of layers of cells. All of them are neurons, and they relay information to one another after the photoreceptors, which are the deepest part of the retina, detect light. And so it's very much kind of a sensory oriented, simplified version of the brain. So I was drawn to that right off the bat, and that's what I wanted to study. I knew nothing about stem cells when I came, so I came here for residency. So after I completed my MD and my PhD in neurosciences, and really I kind of knew what I wanted to do and who I wanted to help before I knew how I could possibly do it. And that were individuals. I'm a pediatric ophthalmologist, so I see a fair number of children and families who have diagnoses where those photoreceptors, those deepest neurons within the retina, degenerate. And they degenerate like midbrain dopaminergic neurons degenerate in Parkinson's disease, or other cells degenerate in other parts of the central nervous system that cause other devastating diseases like Alzheimer. So there's nothing you can do for these patients. Essentially, they start to miss a very critical piece, probably the apex of the visual pathway. And unlike skin, the central nervous system and the retina, which is part of the central nervous system, has no way to repair itself. So there are no intrinsic stem cells in mammalian or human retinas. So once you lose it, it's gone. And so at that point, you have only a couple of options. One would be to bypass the whole system and try to stimulate, the brain directly and that sort of thing. But it seemed to me that the better part and the more natural thing to do would be to replace those cells once they've died. And these are inexorable processes. And so we're very good at telling patients, kids, and families that you have this disease and you're going to go blind. And there's nothing we can do. I shouldn't say that. The Wisconsin Council for the Blind and Visually Impaired here in Dane County is fantastic, and so they have low vision aids and things like that. But I wanted to be able to offer a little bit more, and that's what I wanted to do on the laboratory side. So I had already kind of identified that, and it was 2 years, when I first came here, it was 2 years after Jamie had made his discovery about embryonic stem cells. And so there's the clay, right? So there is the promise that you have these cell types that have the theoretical capacity to make every cell type in the body, including the ones I'm interested in, which can't make them themselves. But that's a long way to go from something that has a theoretical capacity to make these different cell types to something that is arguably the most complex cell in your entire body, and that's a photoreceptor, rods, cones, those sort of things. So I set my expectations kind of low. I kind of said, well, if I can just get to the first couple of steps to something that kind of looked like retina, then at least I would have something I could study, because there's no other way to study human retina. So we had wonderful people here. I was a biochemist, that's how I was trained, so I knew nothing about cell biology. The stem cell community here took me under their wing, and I learned from folks like Jamie and from Tim and John Odorico. It's just a tremendous collaborative community that allowed me to jumpstart way more than I could have on my own somewhere else. I mean, this really was the only place in the world that could accelerate the technology, especially for a novice like myself, at the pace that it occurred. So, I'm trying to work with what's called developmental biology, trying to coerce these cells, which can become anything, which is great, but also daunting. So how do you keep them, if I want them to become a photoreceptor cell, which pound for pound is a very tiny part of your body, right? So how do I get enough of these from this source? And along the way, no offense to Tim Camp, I don't want anything beating in it, right? I don't want any beating heart cells in the middle of my photoreceptors. I don't want any skin. I don't want any of these sort of things, because I need something that's a population that I can learn from and that hopefully I can put back into a patient someday, and nobody wants that in their eye. So it's a very careful series of experiments that we do to try to take what we learned from the development of the retina and apply that to these cells. And we went a long way, and we were able to see things that were retina-like, but they were in a sea of things we didn't want. And so the eureka moment for us was Jason Meyer, who was my first postdoc in my lab. We had a couple of ways of determining relatively quickly whether you were a rudimentary or early retinal cell. So not a photoreceptor yet, but an early retinal cell. And what we found was that by growing them in 3D structures, so as opposed to flat on a plate, growing them actually in a three-dimensional structure, which is how retinas normally grow, that they would separate themselves. Again, this wasn't brilliance, this was serendipity. They would separate themselves from other growing structures that from the central nervous system. So we were able then to find the diamonds in the rough and pull them out. And then over the course of the next couple of years, we were able to find ways to really accentuate and make large amounts of those early retinal, three-dimensional structures. And then over the course of the next couple of years, we're able to coerce them to form retinal tissues. So they would separate out, they would make the photoreceptors, all the different layers, cones, rods, and then we spent a good deal of time characterizing them and pushing them to see that they're really authentic. So are these really photoreceptors that can detect light and come in all the different flavors that they should? We compare them to real human photoreceptors because we wanted something that was really authentic. Close only counts in horseshoes and hand grenades, and it wasn't going to count in photoreceptors either. So we needed to make sure that they were authentic. And so we were thrilled to see that that was indeed the case. So the eureka moment was when we were able to find these large quantities and be able to coerce these cells to become what we wanted them to become, and to become pure colonies of these photoreceptor cells. And so what we hope to do now, and what we've been able to do, is start a company that is able to take this technology and take the resources necessary to move it towards a clinical trial, to be able to test whether or not, in fact, it can form connections with a diseased human retina. And that's really, for the next 20 years, that's the challenge that we have. I'm amazed that we are so good at cell manufacture right now. We can make spare parts for the retina, for other tissues as well. Now the next frontier is, how do we get those to interface, putting new wine into old wineskins? How do we get them to talk to one another to really restore vision? In some respects, I think that there's a probabilistic chance that that will happen at some level, but then we're going to need to improve that to really make meaningful changes in people's lives. So that's what I see, and it's just a wonderful position that we're in right now, that we have the tools to be able to address that next frontier.
Howdy, I'm Dave Brady. I'm one of the members of the Thompson group. And I think I'll answer your questions, Penny, in reverse order. So for me, getting involved in science, I started out actually in business. As an undergraduate, my first year I was a business major. I always loved living things, and I was fascinated by living things, but I didn't think it was practical as far as like a career is concerned. And I had a friend who was a bio major, and he was taking a molecular biology course. And so I would pepper him with questions about what he was learning in his lectures every day. And finally, one day he looks at me and says, "Why aren't you in biology? You love this. Why are you in business?" And that like broke something in me. It gave me permission to follow my curiosity, and it gave me permission to explore that space. So that's what I did. I became involved in stem cells, or I should say interested in stem cells, really after sort of the IPS technology revolution. So if you're not familiar with that, IPS just means induced pluripotent stem cell, and that's a technology that lets scientists take any living cell, essentially, and convert it into a stem cell. And so that the power of that means you can make stem cells from anything or anyone. And that sort of just shook me and made me very curious and wanting to learn more about stem cells. So after graduate school, I became part of Jamie's group. And the work we're doing right now is involved in artery engineering. And we got interested in arteries or blood vessels for two reasons. The first one is vascular disease is the #1 killer worldwide, not only in developed countries, but also in underdeveloped countries. It's a growing problem. So we spend about $500 billion per year treating cardiovascular diseases, and over the next 20 years, that'll grow to $1 trillion. So unlike other diseases that we see shrinking rates and improved outcomes, with cardiovascular disease, it's just getting worse. So that was a major driver. And then the second reason why we wanted to start studying the vascular system is it's important to have mastery over that system if you're going to really entertain the notion of developing organs or large tissues. Because you can imagine if you just have a little nubbin of tissue, then it doesn't necessarily need a blood supply to get its nutrients and to get rid of waste. But as soon as you go much larger than a tiny little nubbin of cells, you need vascularization, or you can't grow much. You can't get very big. And so understanding and controlling the vascular system is this really important prerequisite to all other large organs. So at the moment, what we're interested in is applying stem cell technology to treat people with vascular disease, different approaches to engineer arteries, and hopefully eventually use those in clinical applications. As far as the eureka moment, for me, I think the eureka moment has sort of evolved over time, and it's really been sort of something what David was talking about, the realization that you can make very specific cell types in a dish. So for example, for years, we've known how to culture different types of vascular cells, but in particular endothelial cells. So these are the cells that are the innermost layer of blood vessels. But the cells that you find in a dish, that you can grow in a dish, were generic. Like, they weren't terribly specific to a particular type of blood vessel. But over the last few years, 3, 4 really, we made great strides in being able to clearly specify particular types of vascular cells, not just something that's generic, but something that is very well specified to particular types of blood vessels. And that was surprising to me, that we could make sort of a leap like that. And now the hope is we can take those very specific cells and apply them to all sorts of different types of vascular diseases.
Excellent, thank you. Oh, my God. So we're here, panel, celebrating 20 years since the initial discovery. I'm going to ask you a couple questions, and then we'll reach out to the audience. We'll be thinking about your questions as well. So what do you see in the next 20 years? What are you excited about in this area? Research and development.
So that's obviously a very big question.
Well, yeah. In 50 words or less.
That's probably I need to give back some of my time. I think in the next 20 years, we've been talking about cells as individual cells. We use that terminology, but as both the Dave referred to, what we wind up really wanting to do is to make a higher order structure. We want to make an artery, a vessel. We would like to make a functional visual unit, ultimately hooked up to the nervous system to convey that message to the brain. What what I needed to do in my project was just not take these progenitor cells, but I needed to actually make them three-dimensional also. So there's a theme here between the three of us that I think is what's going to drive this whole field for the next 20 years. And that is the recreation of anatomically and physiologically functional tissue. That would be a tremendous contribution. Diseased organs could be replaced for any of a number of organs, hopefully. I think that is not a simple task by any stretch of the imagination. Look how long it took us to understand from growing human embryonic stem cells, the very discovery that Jamie Thompson made, to where we are today. And that's with a fair number of technologies, that is, different types of cells in the clinic, but those are mostly single cells. And what we need to do is, within our universities and with our partnerships with industry, is to drive that innovation of making these higher order structures, drive this technology into the clinic so we can test it. In testing it, we'll know how we need to make it better, and then we can get it to patients. So that's a little more than 50 words, but I'm going to pass this on.
Excellent. David?
Okay, so I'm pretty clear on where I think that where we need to be in the next 20 years or so, or where we're going to see most of the advances. And I'll say from the onset that just like we were 20 years ago ahead of the curve in making the spare parts, I think we're also here at the University of Wisconsin ahead of the curve in this next phase, and that is interfacing stem cell biology with other disciplines. So building off of what Lynn said, in order to make the cells that we make, the spare parts, interface better and actually function better, we have to look into patterning. So how are we going to pattern these cells? How are we going to mature them? How are we going to prepare the disease tissue to accept these new cells? And so therein lies our increasingly intense interactions with, say, engineering. So nanotechnology, folks, medicinal chemists, people that are looking at small molecules that might promote outgrowth of cells and reconnection or synaptic formation in the central nervous system. We have a project, we've had a couple of ongoing projects with our tremendous biomedical engineers, who, by the way, are stem cell engineers. So one of the first universities to ever recognize this. And so we have a number of our biomedical engineers who specifically work in the field of stem cells. So we've worked with Sarah Gong and Jack Ma, who work on computer chips and the ways to be able to manufacture things on a nano scale or a micro scale. Well, essentially what we want to take is our cells, and they exist in the retina in a very specific mosaic, right? So right now we can just kind of put them in there and they're kind of haphazard, and we can hope that a few of them line up the right way and connect the right way. But what if we can get them to stand up like little tin soldiers so that they're actually in the correct mosaic, like a patch? Well, I have no idea how to do that. But Sarah Gong knows all about biomaterials and what degrades, what doesn't, what's compatible with the human in different compartments. Jack Ma knows how to make things very precisely on a very tiny scale and to do it in an economically reasonable way. So then we get together, because this is a highly collaborative university and everybody likes to work with everybody else and expand what we can do. So, and similarly, we might need a few growth factors or other kind of molecules there to support the system, make it survive, make it grow and thrive. So working with medicinal chemists to create molecules that might help become some sort of a glue between the new cells and the old. So again, we've fairly quickly, in the grand scheme of things, not if you're suffering from a horrible disease, of course, or going blind, but from a scientific medical standpoint, we've moved fairly quickly at having the tools, having the spare parts to the airplane. Now we got to assemble the airplane, and that's not a trivial thing to do, and that's going to require people with much more expertise than just biology.
So I totally agree about this important inflection from cells to tissues that's happening right now. And the other panelists are exactly right, that it's going to require multiple disciplines coming together. The one last thing I want to throw in is there's obvious applications here for treating diseases and also for drug discovery. But I would say over the next 20 years, we'll also see stem cell applications outside of those obvious interventions. One, for example, is in toxicology. So the vast majority of chemicals, both natural and man-made chemicals that we encounter on a daily basis, are not well tested. And the reason for that is it costs about $1 million to thoroughly test a chemical, and that's usually done in mice. But mice are not humans. And so if you look at the number of molecules that behave the same way from one species to the other, it's about 60%. So you have a coin toss of a chance of what you find in the mice is really going to be applicable to what you find in a human. So I think with these complex structures that other folks are talking about, we'll now be able to very cheaply screen all sorts of compounds and have a much better sense of their potential toxicology or their safety, specifically in a human system.
Before we throw it to the audience, do some of you have questions? I'm just curious. Are there a lot of questions in the audience? Have some. Okay, hang on to it. We're going to ask one more question and then we'll come back. So we've talked about sort of the promise of the next 20 years and what needs to happen. What are the challenges that you all see? Another real small question.
But it's a really important question. So here's the challenge. The start of the challenge. This is a larger bucket challenge. And that is how medicines are assessed and approved by governments, not just in this country, but worldwide. Is this important? Absolutely, it's important. The Food and Drug Administration in this country, the European Medicines Association in Europe, these are bodies that regulate how we make medicines, how we say they do anything, or they're efficacious. And this testing takes a long time and it's very expensive. If you take the long view and look what's happened historically in the field with the Food and Drug Administration in the U.S. in particular, those people who are those products that are pioneering products, and stem cell technology, without a doubt, is a pioneering type of new medicine, cellular medicine. And so its path is going to be longer, and its path has been longer. The National Institutes of Health, David and I spoke at that meeting in December 2017, was actually bemoaning the fact that the path was too long, and they have instituted programs to accelerate the path. But I think that a fundamental element is having technologies get through the Center for Biologics at the FDA, and by get through I mean become approved, shown that they're efficacious, have them assessed by experts in the field, and the FDA say, yes, this is approved, this may be available to patients now. And that is most likely going to require a predecessor, and that predecessor, I believe, is going to be a progenitor human cell, probably not a human embryonic stem cell or an induced pluripotent stem cell. And from that, folks in that field that are directly working with that will benefit. They'll be able to go faster. So I would predict it's a challenge because these medicines are complex medicines. We're talking about having them have a structure that's even more complex. There are a lot of challenges, but there needs to be some model, and from that, new regulations will be written. I'll just end by saying, when I started and founded a small company so that we could actually do clinical trials, the guidance documents that were available for us from the Food and Drug Administration were not relevant to what we were doing at all. So we needed to write and develop, even the clinical trial design needed to be unique. And I sincerely hope that that will help others in some form.
So I think already said that I think that the biggest challenge is installation. So I look at it in two groups. Cell manufacture, cell installation. Installation is still a big challenge. So as not to repeat myself on that, I'll bring up another challenge that is nothing to do with transplantation, but is also a very important application of stem cell technology. Also kind of that Dave mentioned as well, and that is things that we can do with these cells without putting them back into a patient that still have direct relevance to treatment of disease. So for instance, I mentioned before that animals are not the same as humans, of course, and the human retina is different in many respects than animal retinas, and yet that's what people usually use to test medicines. So not just toxicology, which is important for safety, but if you're developing a new molecule, so a new drug, a pharmacologic agent, or a gene therapy, you have to, you need something to test that on. And so if you can make actual human retinas or human tissues from patients who have that disease, you have a very unique and very potentially powerful model system to advance gene therapies or other pharmacologic agents. That's not going to help patients when their cells are gone because there's no longer any cell left for the drug to act on or for a gene to get plugged into. But if you're earlier in the disease, then we can accelerate the development of those products and to determine, hopefully, whether or not they're going to work using a more relevant system. So what's the challenge? The challenge is being able to take that idea, which plays well and on small scales can be done, but making those organs, those organoids that we can do so well now, but to make them on a very repeated, so they all are very similar to one another, and to do that at a high enough scale where it makes sense to screen lots of things and to accelerate the process. So converting our systems into something that is highly reproducible and scalable, also bringing in our bioengineering, I'm really plugging the bioengineers here, our bioengineering colleagues to be able to do this, I think is going to be able to really expand what we can do with stem cell technology, both in terms of cell replacement as well as modeling to accelerate more established forms of therapeutics.
Awesome. I don't have much more to add. For me, over the next 20 years, the biggest challenges are functionality, efficacy, as been previously discussed, and then also safety. So it's, of course, super important that whatever cell-based therapy you're pursuing, that the cells don't do any additional harm, give rise to some sort of cancer or otherwise cause problems for whatever the condition you're trying to treat. And so those, I think, are the two things that we worry about the most. Are cells safe? And there's lots of different ways to try to figure that out. And then are they functional enough to do what we're hoping that they can do?
So we have more questions, but we thought we'd go to the audience for a little bit. And if you all had any questions? There was one over here, I think, and microphone runner.
Yes, thank you. So like 13 years ago, I had sudden hearing loss in my left ear overnight, and they determined that it was most likely viral, and what it did is damage the nerves on the nervous side of the hearing system. Now I'm, that much older, and I'm taking drops for glaucoma, and so I've got some more nerves that are degenerating. So my question is, does the stem cell apply to the nervous system? I mean, is it just another application to apply it to nerves so that I can look forward to hearing again someday and in stereo?
I'll take this one.
Thank you.
So what you're talking about is our cochlear hair cells, right? So it's if it was viral and you had acute hearing loss, are you sure it wasn't inner ear? That it wasn't the cochlea? So it was an encephalitis? Okay, I would just go back and figure out exactly what, because it's important, right? So these things are relevant now, because just like I'll use an analogy. So just like if you have a car and you want to know what's not fixed, what needs to be fixed, right? So if your pistons are perfectly fine, but it's your carburetor that's bad, you don't want to put new pistons in, right? So it is important. So when you say nerves, it's not specific enough, right? What type of nerve do you really, or cells is it that's damaged? Okay, so a cochlear hair cell is much different than a cortical neuron. To answer your question is all those cells can be made, right? The question is, but can they be installed? So we have, right, so cochlear hair cells are actually, they can be made. They'll stand up in a dish. You can get them to respond to sound waves, and they'll generate an electrical signal from them. It's really quite amazing. So you can do that, and people are working to be able to implant those cells. Now, the other thing about that is you have cochlear implants, so there's also engineers, again, that can perhaps do a little better than the cell biologists, at least right now. With regard to cortical neurons, of course, the more complex system that you're dealing with, the more difficult it is to replace that function. So when you're talking about higher order cortical function, we're a long way away from being able to do anything about that. So, for instance, Alzheimer would be a very, very difficult thing to reverse, because you're talking about all sorts of cognitive functional things that have to be connected, that are developed over time. And so again, we have to, unlike the shyster stem cell clinics you see that will say they can do everything but not explain how they can do it, because it's some sort of magic, and that's really what they're trying to sell you. You have to really think about the biology, because it's all rooted. This is not magic. It's rooted in developmental biology and the way your body works. So then, with regard to glaucoma, which is totally different, by the way, that's apples and oranges. So what you had, the viral infection you had that affected your hearing, is totally different from glaucoma, which we don't necessarily know how it starts or why some people get it and don't get it. That affects a cell type in the retina called the ganglion cell. Now, the reason I'm going to go into this is because it, again, tells you why it's important to know what you're trying to replace. Ganglion cell is also in the retina. So why am I not right now trying to replace those? The difference is a photoreceptor cell exists in the very deep recesses of the retina, and it makes one connection to an immediate neighbor within a micron away. So if you get them in the right spot, which is actually easy to do surgically, we're in that space all the time, then you just have to bump into your neighbor. They then feed downstream to other cells, and then ultimately they get to a ganglion cell. That ganglion cell, there's 1.2 million of them in your retina. They have to go all the way down to your optic nerve, hang a right, go all the way into your brain, and find a precise target in the lateral genicular nucleus in your brain. So replacing that is a tall order, right? That's sort of like me. I'm a tall dude, and so if I have a spinal cord injury, you got to get something in my spinal cord, and then it's got to find my left big toe, right? That is a hard thing to do. So replacing a retinal cell you can't say that generically. The odds of replacing a photoreceptor are much, much higher than replacing a ganglion cell. So again, I go into the detail just so that people get a sense for it's not the brain or the retina or even the skin or the artery. It's what specific cell and what groupings of cells do you need to have replaced? Can we make them? Can we install them?
Thank you. It's amazing, and I know I so appreciate. I bet you do too. Complexity and the networking that happens with all of this work and how it comes together, and when a discovery happens, and then it's 20 years later, and why aren't miracles happening every day out of that discovery? It's a long road that needs a lot of patience and dedication. You have another question? Do a couple questions. Oh, they're popping up. Microphone.
A much less technical question. How has government policy affected either where you've been in the last 20 years or where you may go in the next 20 years? Like funding, regulation, administration, and does it matter really who's in power?
Okay, that's a really critically important question, because none of us would be up here today without funding from our government, the National Institutes of Health, the Department of Defense. This is a fabulous thing we can appreciate in this country. So, with regard to what is promoting specifically stem cell technologies, I can tell you that most recently, the President Obama, and this was in December of 2016, signed something called the 21st Century Cures Act. And this is an incredible foundation piece of legislation that put into law guidelines for the FDA to be able to have a way to accelerate certain types of technologies through something called the Regenerative Medicine Advanced Therapy Designation. And this helps biotech companies, pharmaceutical companies that are developing new cell-based medicines or cellular-derived medicines get these products assessed quicker, depending upon the data, of course, if the data warrants this, through the FDA and to patients. I can tell you that the technology that was developed here in Madison in my laboratory was the 4th technology in the United States to be granted regenerative medicine advanced therapy. To my knowledge, it is the only product in the Midwest, but there are less than 2 handfuls of products that have gotten that designation to date, but this is important. Worldwide, I'm also going to comment that Japan has done a very interesting, I won't call it experiment, but in a sense they are experimenting. So whereas in the United States, you do need to go through the full regulatory pathway of doing clinical trials through a phase 1 focused on safety, what's called a phase 2, a larger study also focused on safety, to a phase 3 and maybe 2 phase 3 studies to determine if your product does what you hope it does, that is efficacious, has a strong effect, okay? And then you may provide it to patients, commercialize it. In Japan, the following change, that variation of that, is that if you do a phase 1 study and you show a cell-based product is safe in this initial study with very small patients, you may start to commercialize it. You can immediately get it to patients. You have 7 years to do the full series of the next series of studies, whatever those might be, depending upon your cell-based therapy, to show that it is efficacious. If you don't show it's efficacious by that period of time, you may no longer sell it. But this is a way to encourage, to bring support monies into smaller companies that are developing technologies. So overall, I would predict that we'll see more creative legislation, more support of these types of new medicines, because I anticipate they're going to be very potent. The oncology field is showing that these types of cell-based medicines are very potent.
Thank you, Lynn. Another question from the audience. This side of the house hasn't had a chance yet.
Hi, Don. Recognize another questioner. My question is pretty simple. I'm just wondering what the status is of the clinical trial process with using human.
The question was the status of clinical trials?
Clinical trials.
Stem cell research? Yeah, that'd be clinical trials with human subject. Yep.
So in many areas, the eye is fortunate in that it's on the forefront of a lot of the clinical trials that are going on right now, and the reason for that are a number. One is that you've got 2 eyes, so there's one you can play with, which is I'm not kidding you, right?
That's right.
It's also an encapsulated organ. This kind of goes into the thought process here because it all comes down to safety. It comes down to what's your risk-benefit ratio. If you're doing something in the middle of your brain and putting cells in there and something goes wrong, you can't necessarily get them back out again. So there's a high risk there. So in the eye, it's sort of like a mini brain, but it's forward. It's easy to access. The surgical techniques to get the cells in place are straightforward. We don't need new instrumentation, which all of this raises the cost and the duration of these studies. Because if you need new instruments to deliver your cells, those instruments also have to be part of a trial. So if you can bypass that, then you just reduce the cost and made it more desirable for a company to pick up the cost of a clinical trial. So the eye is that way. The surgical techniques are straightforward to work underneath the retina. The instrumentation is generally there. You have 2 eyes. The eye itself is relatively encapsulated, so if something goes wrong, you can, worst case scenario, remove an eye, You can't remove your brain, but you can remove an eye. And the other thing too is that our techniques for monitoring what those cells are doing and how they're functioning is very sophisticated. I don't need to put you into a PET scanner or do an MRI every week to see what the cells are doing. I can just dilate your pupil and take a look. And so it's non-invasive. And then we can do a whole host of studies to determine if the cells are working, what they structurally look like, all non-invasively. So the ability to monitor in both the structure and the function of the tissue in a non-invasive way also makes it straightforward. So what does that bring us to? It brings us to an awful lot of studies right now, clinical trials in humans, looking at a particular cell type that is the helper cell for photoreceptors, which is called the retinal pigment epithelial cell. It's the first cell that's damaged in macular degeneration, but it's not why you go blind. You go blind because the cells that it subserves, the photoreceptors, die secondary to the loss of the RPE. RPE is relatively easy to make. It's pigmented, it looks different, you can cut it out, and so it's well ahead of the curve in terms of being put into patients. It's been shown that it's now, I think there's more than a half dozen clinical trials, many patients have been treated, and it's been shown to be safe. Essentially, it doesn't reverse much in terms of blindness because it lacks that photoreceptor cell. But it's taught us a lot about how to deliver the cells, the safety level, and it's also informed the FDA a lot in terms, so it's paved the way for folks to come up they're coming up behind them with other cell types like photoreceptors. So we kind of have a blueprint in terms of how to go forward. So we're hoping to build off of that and get better results long term. But I think that, again, the FDA is not our enemy here. The FDA is, they're there to make sure that we first do no harm. We don't want to do any harm, but I don't have the experience of doing 100 clinical trials. I don't know what all of the pitfalls could be for a patient. The FDA, that's their job. They know things that I would never know in 1,000 lifetimes. It's their job to apply that to make sure we do what we want to do, which is to help patients and not hurt them. They very much want these therapies to go forward too, but if we make a misstep and somebody gets hurt, it will set the field back a decade. That's what happened with gene therapy, and it will happen with cell-based therapy too, and we'll all be in just in a holding pattern until we figure out how did this all happen. So, being a expeditious turtle, so being a focused, the hair is not what you want. You don't want somebody that's some cowgirl that's going to throw things in your eye and say, well, we'll let the cells sort it out later. But you want to be very directed, and you want to have a purpose and a goal, and you want to get there as quickly as you can, but as safely as you can too. We're doing nobody any favors by trying to jump the gun and hope that something works when we really don't understand it. Which cell?
Yeah, well,
I'm going to go back to that. They're not the same.
Also, if you want to follow the clinical trials that are happening around the world right now, go to clinicaltrials.gov and type in embryonic stem cell or pluripotent stem cell. They're all listed. What's going on, the data, everything is right. Yeah, you're welcome.
We have time for one or two more questions.
Hi.
There's one in the back there.
I have a mic, so I'll ask one. I just wondered more generally for more information about where stem cells are sourced. It's not a loaded question. There's no agenda, but I just was curious. It sounds almost like they're self-propagated now as a medicinal product, or disseminated to each branch of your medicines. I just wondered more, where do they come from, and how do you get them?
Sure. So there are a number of lines that are established, that Jamie established 20 years ago, that are in use heavily around the world. And those are distributed by YCell here in Madison. That's a nonprofit organization. And so those have now, actually I have some stats on them. So Jamie's 5 lines, there's been 5,200 distributions to 2,350 independent investigators at 820 institutions in 45 countries across 6 continents. So they're everywhere. So these are sort of the classical lines. And then with iPS technology, that allows you to make pluripotent stem cells from any sort of donor. And there's many, many, many iPS lines that are available as well.
Thank you. I'm afraid we have to draw it to a close. It was a quick hour, wasn't it? But we want to thank our expeditious turtles. Do we need any final instruction for the audience except to go out and explore?
There are some hands-on activities out in the town hall, so please check it out. And we also have a special